We'll go ahead and get started here. Welcome, everybody. It's good to see that there's quite a few people on here. So today's talk is going to be, what do soil tests tell us? And I guess I should introduce myself first. I'm Ryan Slaughter. I'm an extension educator down in Ross County, Chillicothe, Ohio, if you've ever been through there. Oops, looks like it's progressing on its own. So a couple housekeeping things this morning. There will be a CCA credit that we'll put up at the end of the talk. This session will be recorded and it's available at agcrops.osu.edu. You can find all of these there. Also, there's going to be a couple different features. You can either put stuff in the chat for logistical reasons, addressing the presentation, or if you've got some questions, you can use that Q&A feature. That's one of my favorite little features of these Zoom talks, because we can either address those in that chat area or answer them live. If you got any other questions at the end, you can email Rachel at cochran.474. And then again, with the chat features and poll questions will be there. So and then Sarah put in there, but in the chat also, if you'd like to, you could put in where you're from, either in Ohio or out of Ohio, and what city you're from. It's always kind of neat to see where everybody's tuning in when we do these Zoom talks. So today's speakers, Dr. Jim Ippolito and Dr. Elizabeth Hawkins. Jim will go first, and I will introduce Jim, and then I'll stop sharing. Jim is a professor of environmental soil health. His 30-plus year career is focused on improving environmental soil fertility, chemistry, microbiology, soil health within agriculture, short grass steppe, grazed, burned, and metal contaminated mined land ecosystems. His research program connects the intimate linkages between soil macro and micronutrients, trace and heavy metals, microbiological activity, and soil physical attributes, and how these factors combine to influence ecosystem sustainability, resiliency, food and climate, and environmental security. Jim, welcome.
Thanks, Ryan. Good morning, everyone.
Okay, I stopped share, hopefully.
Okay. Well, it's a pleasure to be here this morning to talk to you about soil health testing and what these tests are actually telling us. And so I think about soil health a lot since I've been in this field for almost, if not my entire career, which is like 35 years. And so I think about soil health and the approach that the NRCS takes and the approach that many of us take now follows this diagram in the top left-hand corner. The principles of soil health, and there used to be five, but now there are four, and so a couple were combined. So the concept is to improve soil health, to minimize disturbance, maximize living roots, maximize soil cover or armor, and then maximize biodiversity. However, you can do that by either introducing livestock if you can, or increasing the number or types of plants or plant families in an agroecosystem. And so we aim to do this in order to see what you see on the right-hand side of the screen. So maximizing cover, having continuous living roots or plants throughout the season or throughout the year, all in order to improve soil health. And you can maybe kind of see soil health in the top right-hand corner and the bottom right-hand corner in terms of aggregate stability and organic matter content in living organisms like roots, for example. And what we strive for is for improving soil health, really. This is the concept behind this talk and this webinar series. And so I really like this definition from Doug Carlin. Doug was instrumental in bringing me into this soil health realm when I was younger in my career. And the definition from Doug Carlin was, or is, the continued capacity of soil to function as a vital living ecosystem that sustains life, essentially. When I think about soil health and what I'm going to share with you this morning, I I like to point out that soil health incorporates a number of different constituents and sort of a holistic view of ecosystems or agroecosystems. And in not just my opinion, but the opinion of a lot of people that work in soil health, in the soil health arena, that when you quantify soil health, you really should be quantifying soil physical, chemical, and biological attributes. So I want you to keep that in mind as I move forward in this slide deck. So what you see on the right is really what we want. And what we don't want are what you see here in these three pictures. And these are all relatively recent pictures. And so if you're living in a system that is or similar to any one of these three pictures and you want to make a change, how do you guide yourself towards a positive change? And the way you do that is you need to quantify your ecosystem or your agroecosystem and quantify it over time. And so we can use soil health testing to do this. So there's likely more than, I know there's more than just three soil health tests that people can use. The three that we typically use in my research program at The Ohio State University are listed here. The first one is called the Soil Management Assessment Framework. And this is a soil health testing framework that's primarily used by scientists like myself. All right, so this is not a test that's used by everyday people or laboratories, private laboratories. The second soil health test framework or protocol is called the Cornell Assessment of Soil Health. And this actually can be used by not only scientists, but it's used by some laboratories for soil health testing, including the laboratory at Cornell. And then the third test is called the Haney test. And sometimes this is referred to as the Haney soil health test. And I'm going to give you my opinion about the Haney test. Um, and I don't really consider it a soil health test. I consider it more of a soil biological test. And you'll see why in this slide deck. And I'm not saying it's good or bad. It's actually a really nice test to guide you in your journey. So I want to talk to you a little bit about these first two tests and what they tell you. All right. So this is a slide, and on the left-hand side are the attributes of soil indicators that go into the soil management assessment framework. And on the right are the attributes or indicators of soil health that go into the Cornell assessment of soil health. And I tried to line up these indicators or soil attributes on the left and right so they sort of match each other. So you can see that both of these protocols are quantifying soil physical, chemical and or nutrient and biological attributes of soils. So on the left, on the soil management assessment framework, we quantify, or this test quantifies how dense the soil is. All right, so is it friable, easily workable? You can put a shovel in it, or is it as hard as a rock? It quantifies aggregate stability. Um, this test, if you were to do this test, if any of you wanted to do this type of test at a testing lab, when you collected a soil sample, you need to make sure that it's relatively intact, that you don't destroy the aggregates prior to shipping to the laboratory. So this test is a little sensitive. The soil management assessment framework quantifies what the soil pH is. It quantifies EC, which is a measure of salt concentrations, and it quantifies plant available or extractable phosphorus and potassium. These are the chemical and nutrient indicators. And then in biological, we quantify soil organic carbon. We quantify the amount of carbon that's in microbial tissue, so microbial biomass carbon. This test quantifies something called potentially mineralizable nitrogen. This is the nitrogen that's present in an organic form that can be released as an inorganic form to feed plants. And then it also tests something called beta-glucosidase activity. This is an enzymatic activity that quantifies the ability of microorganisms to degrade easily degradable carbon compounds like cellulose. And then it also quantifies clay content, which is sort of an overarching independent variable in SMAF. All right. So if you switch over to the right-hand side of this diagram, the CASH, Cornell Assessment of Soil Health, follows almost the same protocol. instead of quantifying density, it quantifies something called penetration resistance. So this is when you push in, either you could push in a knife, you could push in a penetrometer, you could push in a shovel, and it measures, or you measure resistance, which is a quantification tool to measure maybe density or compaction layers in soil. It also measures wet aggregate stability, and so those are physical attributes. It quantifies chemical and nutrient attributes, and it quantifies biological attributes that match up pretty well to the soil management assessment framework. And then overarching, it quantifies texture, which encompasses clay. And if you have any questions about any of these indicators, feel free to put them in the chat and we can revisit later. So this is what we do when I'm doing my job at Ohio State University. I have grad students and they're really interested in measuring soil health. And so this was one project that we recently worked on here in Ohio with 11 different sites that you see on the far left-hand side of this table. The table's really busy. And what I want to do is just focus on the overall soil health scores. So what you see on basically the left side of this table is the soil management assessment framework and the overall soil health score. And on the right-hand side is the Cornell assessment of soil health and the overall soil health score. So if you focus on the two columns that are color-coded, the color coding matches how the Cornell assessment of soil health would color code their overall soil health score. So you can see in the bottom right-hand corner how Cornell color codes. And so if you look at the color coding for the overall soil health score in Cornell, Cornell uses a scale from 0 to 100. And you see by and large that most of these colors are light green. In the soil management assessment framework, the scores range from 0 to 1, but if you range them from 0 to 100 and then color coded them, this is what you would see. And most of these are dark green. And so when you look at these two columns of color codes, what you see is that, they're relatively consistent. Most of the color codes are light green in SMAF, in cache, and most of them are dark green. And this tells you something about both of these tests. It tells you that the Cornell assessment of soil health is a little bit more difficult to get a higher score than the SMAF, but they match pretty well, to be honest with you. I'm guessing though, I want to switch gears just a little bit, well, a lot of it, and I want to talk about the Haney test. And I apologize that this table in the top right is a little bit fuzzy. I borrowed this from a testing laboratory. The Haney test looks at three different indicators to derive a soil, they call it the Haney soil health score. It uses the first one, actually it uses three, all right? So the first one is a carbon dioxide burst, and that's what this first row is. The second one is water extractable organic carbon content, and the third is water extractable organic nitrogen. And so if I walk you through how the Haney test works, the first score or the first component of the Haney test is CO2 burst or respiration. It's basically how much respiration or carbon dioxide occurs over a 24-hour period. You take a dry soil, you re-wet it, and then you capture the carbon dioxide that's given off over 24 hours. And that's the value or the number that you see in the top right, the 109.1. Essentially, the more carbon dioxide, the more microbial activity. The second indicator is water extractable organic carbon content in the Haney test. And this measures the amount of organic carbon that's readily available for microorganisms to consume or to eat to help them respire. And the greater the number, the greater amount of, quote, food that microorganisms have. This is essentially how this works. The other part of water extractable organic carbon that's relatively important is that, again, this is the food for microorganisms that is coming from the soil organic matter phase. And if you know anything about soil organic matter, it's not one item, it's a continuum from something that you can physically see with your eye to something that you can't see at all. And this water extractable organic carbon comes from some phase across the spectrum, right? And so what you want is a higher number. You want more food for microorganisms. The third value that goes into the Haney test is the water extractable organic nitrogen. And this is the amount of nitrogen that's present for microorganisms to utilize to create amino acids, proteins, and enzymes so they can do their job to effectively force nutrient cycling and turnover in soils. And so you want this number to be as high as possible as well. I failed to mention where most of these numbers fall. And if we want to go back and revisit this during the Q&A section, we can. So this is what happens. So here is a Haney soil health test score or test. There's a lot of information on this, but the soil health score itself, which you see here, 16.9, is derived from what we just went through. And so if you look at the box and then you look at the number, how do we get to that number? This is what testing labs do. They take these three numbers and they plug it into this equation that was derived by Rick Haney. The Haney soil health test score is a measure of how much CO2 is given off, plus the water extractable organic carbon content, plus the water extractable organic nitrogen content divided by some values. And if you take those numbers in that table and do the math and continue to do the math, you finally end up with 16.9. Now, this number to me doesn't mean a lot, to be honest with you. So the Haney soil health test scores typically range from between 0 and 50. 16.9 tells you that you're somewhere in the middle to low range. But what could be useful for using this test for some of you that may use this test is it tells you where you roughly are in a system, just like the cache and the SMAF. And if you use this test over time, it can tell you which direction you're moving. The one part or two parts that are missing from the soil, and it's called the soil health score, but it's only attributing, so quote, soil health to microorganism activity. And it's not attributing soil health to physical or chemical attributes. And so I would call this a soil biological health test score, is really what I would call it. One last thing. So I have a lot of producers that come to me and say, okay, well, what is the Haney soil health test score telling me? And I tell them, well, I think it's a biological health test score, but let me run a project to see if I can figure out, or we can figure out what else it's telling us. And so what we did was we had a project where we used the soil management assessment framework, which are these three circles on the left. And we pulled soil samples from 8 different locations. And then we ran those soils through the soil management assessment framework. And then we also ran those soils through the Haney soil health test. And we did something called correlation analysis. And this analysis was a little complicated, but I'll try to tell you what this is telling us. So we tried to correlate every single facet of SMAF to every single facet of the Haney test. The larger the number on any one of these arrows, the greater the correlation. And if you see a negative value, the larger the negative value, the greater negative correlation. So what this tells you is that if you look at these numbers from the left 3 bubbles to the right-hand side or to the center, the values are really, really low. And if you look at the values that are pointing towards the Haney soil health score, the largest number is coming from the CO2 burst, really. And so when I tell people what we found, I say, well, there's really little correlation to any chemical or physical attributes that are remotely used in the Haney soil health score. And so I tell them that most of the Haney soil health score is coming from the CO2 burst, which is an easy test to run in the laboratory. It's a little costly, but it's not that costly. It might cost you like $30 to run. So let me skip this slide, and I want to go to the last one so I don't take up all this time. When you run a soil health test, it doesn't matter which one you really run, but you should be consistent when you run these tests. You should be looking at whatever test you're using, pulling soil samples every year or every other year or every third year at the same time of the year and run the same test so you know where you start and where you're going. And I'll stop there and turn it over to Elizabeth. Thank you.
Okay, thanks, Jim. Elizabeth, before you get started, let me do a quick introduction for you. Dr. Elizabeth Hawkins is an associate professor and agronomic systems specialist. Oh, my screen just disappeared.
Oh.
Sorry. Dr. Hawkins' research interests include using on-farm research to identify methods to increase farm profits, including improved crop yields, better management of resources, and the employment of effective precision agricultural technologies. She is using on-farm research to better understand how to make precision technologies like site-specific and prescriptive agriculture work, both agronomically and economically. Welcome, Elizabeth.
Good morning. I'm really excited to be joining you guys this morning to talk about soil health. Jim gave a really great lead in to where we're going to go. I'm going to shift gears a little bit. I'm also going to talk about some tests, but I'm going to talk about how we've done some on-farm research with those tests to answer some farmer questions. So starting off, a lot of the information I'm going to talk about today is gained from information that we gathered through our eFields on-farm research program. Starting in 2020, we were approached by a group of farmers who had specific questions about soil health tests and their usability at the farm level. And so we designed a protocol around that approach. The core question we were looking for was, are tests for soil health reliable for benchmarking soil health on the farm at the field level? And they came to us with several requirements for them to want to be adopting these tests and using them. One of the primary goals was to identify a test that was affordable. We know some of these are quite costly, and we really wanted to get an ROI from the information that we're gathering and be able to run these tests on a regular basis without breaking the bank. Then we also needed them to be reliable and repeatable. We know when we're sampling for soil characteristics, it can be really difficult. There's a lot of noise in that field. We wanted to make sure that we weren't adding additional noise by the complexity of the test that they're running in the lab. So one that is going to be pretty stable over time, that has the ability to detect changes that are a result of our management changes. We're really key to filling the need of benchmarking our soil health, because at the end of the day, we want to see if what we're doing on the farm is changing those values, not is the way the test is being run in the lab or the way we're collecting samples changing those results. So a group of researchers worked with those farmers to come up with a study design that was going to help us answer those questions. So we went with a statewide sampling approach. So since 2020, we've sampled 650 fields across Ohio, and we've covered 55 of the 88 counties. To begin to understand the differences between the sample results and some of those management practices of interest, we collected data that helped us zone in on what environments these samples were coming from. And we specifically looked at those conservation management practices. We wanted to be able to compare the impacts of long-term no-till to conventional tillage, see what cover crops do as we enter the system and how they impact soil health, and then specifically wanted to look at, did manure impact soil health as well? So across those 650 fields, they were sprinkled across all of those management practice buckets, as I'll call them. And then we were able to do some things with the data that I'll show later that helped us answer some additional questions to just drilling down on what the best soil health test was. At the individual field level, this is what our sampling strategy looked like. We did take on a whole field sampling strategy. Each sample that we submitted to the lab was an aggregated sample consisting of 10 to 15 cores, and then those were mixed. In the early years, we did test depth. So if you're interested in how depth impacted our results, we could talk about that. But today I'm only going to talk about the 0 to 8-inch depth. Our ending recommendation on depth was that 0 to 8 is a great sample depth to pull because it's going to match with your fertility samples, which simplifies things. And a lot of universities, Ohio State included, are publishing their data based on that 0 to 8-inch depth. So it'll help your results match up with some of the resources you can use through the universities to benchmark your results against others. So we shipped them off to the lab, and the analyses that we went with was first a routine soil test. So looking at pH, organic matter, which we'll talk about a lot, and then also all of the Malic-3 extractable nutrients. So what you're commonly going to get back when you send out a soil test just for your management of fertility overall. The soil health tests that we focused on for this study were POC, which is a little bit of a deeper, more specific look at that organic matter component in your soil. It focuses a lot more on the active carbon in your soil. So you'll see the differences. I'm really going to hone in on our data slides about the differences between organic matter in that simple loss on ignition test and then this slightly more complex but also informative POC test. We also looked at respiration, which is that critical piece of the Haney test that Jim talked about earlier. Protein, and then we included a physical test of wet aggregate stability as well. So the big question, what did we learn from 650 fields of soil test data? And so I want to kick off this discussion with going with what our best practices were that we identified for if you want to pick up soil health testing as part of your management strategy. And the first and most important thing is to set your goals up front. All of these tests are a little bit different in what they're going to tell you and on what time scale they're going to give you information. So understanding what questions you want to answer is going to be critical to picking the correct test that's going to achieve those goals. And then Jim closed on this, but we think it's critically important. We're always going to recommend that you compare the changes in soil health over time within the same field and maybe even within subzones within those fields. We did see, I'll mention here in a minute again, we did see a lot of correlations between some soil properties that influenced soil health tests and your starting points and what you were able to achieve. So there is going to be some just physical soil characteristics that are going to impact the range of values that you might see in these soil tests. But really, I mean, those two things, if you focus on those, the test you select and how you use it is really going to do a better job of answering those questions that you're looking to answer. So because goal setting is so critical in my mind for this, I wanted to spend a few moments discussing that specifically. So back in 2022, we took the first years of our data collection from the eFields project and combined it with some other researchers at OSU to create a data set that had over 2,000 fields of very similar soil health data to what we're talking about. And what we were able to do was take that data and identify for you and summarize some trends that you might see across those different soil types in Ohio. Specifically, we saw that texture and CEC of the soil really impacts some of these soil health test results. So when you're making comparisons, keeping in mind if soils you're trying to compare are very different in terms of texture or CEC, it's going to be difficult or nearly impossible to make a meaningful comparison between those fields. So in this particular data set, all of the tables for benchmarking are broken out by those characteristics, so you can compare apples to apples across soils. Again, this is really why we do recommend at the end of the day, once you set that goal for your farm, you're comparing your fields over time. But this resource, the link there on the left, can be really helpful for understanding your baseline and where you fall on that continuum of soils in Ohio and determine what you think good is going to look like for you on your farm. So the nitty-gritty of some of these tests, these are the key takeaways that we pulled out from what we learned with our experience looking at these test results across a wide range of soils. Organic matter is one that really came out for us as a top contender for, as you can see here, a lot of reasons. Organic matter is going to be a fairly inexpensive test, and in many cases, it's included in your routine analysis. So if you're soil testing every 3 or 4 years, you're automatically getting that value, and you can begin to draw some conclusions on soil health from there. I'll show our data here in a few moments, but we did see a positive yield correlation in both corn and soybean with increased organic matter. So if your goal is to increase yields through soil health, organic matter is a good test to run because we know that if we can increase that value, if we're pushing it forward, we have that correlation to yield that we can lean back to. The biggest drawback for organic matter is that it's going to be less sensitive for detecting those changes over time. There's a lot that goes into building organic matter. We know it's a slow process, so we shouldn't expect to see changes in those soil tests happen very quickly. So if you're not a patient person, that might be a reason that you start looking at these next several tests. Across the board, when we looked at POC, wet aggregate stability, and other tests, so respiration, protein, and I think some of the more complex tests that we didn't specifically test in this study can fall into this category. They are all across the board more expensive. For us, POC and then our respiration and protein tests were fairly similar in cost. Wet aggregate stability, because it is a physical test that's labor intensive, was the most expensive of the tests that we ran. POC specifically is the other one that when all of our data I'm going to focus on, because I think if organic matter doesn't do it for you, POC should be the next candidate that you consider. The reason that I would suggest you go with this over organic matter, the primary reason is that it's going to be more sensitive to changes in that value as you change your management. I'll show that data here in a moment, but we did see that we saw changes in soil health if we're using POC as that marker more quickly than we did with organic matter. Very similar to organic matter, we did see a positive yield trend with corn and soybean as we increased our POC values. Jim touched on this with the wet aggregate stability and then the other tests. They can be very sensitive to the way that that sample is collected and handled, which I think is going to be the next primary drawback. And the reason that I would, in a lot of cases, steer you away from these tests if you're wanting to look at changes over time. Jim mentioned specifically with wet aggregate stability that you need to send those samples in with the aggregates as intact as possible. And that is absolutely true. If you're trying to add this on as just another test with the regular sample you're sending in, it conflicts with how we traditionally collect those samples. So when you mix those aggregates together to create your main sample, you're breaking up those aggregates and you're going to reduce the quality of the results you're going to get from the wet aggregate stability test. We've also seen that because it's a physical test that someone has to run, the results can be pretty inconsistent between labs, can be inconsistent between years depending on moisture content. And so this test in general can introduce a lot of noise that's going to make it difficult to interpret that test and draw conclusions over time. It's similar for the respiration test specifically. That one is pretty time sensitive when you get it to the lab. It is, again, looking at that biological component of the soil in some cases. And if that sits on a shelf for a while, the results might not be telling you exactly what you're hoping they are. So this is our breakdown of what we learned. Experiences may vary, but across all of our data, these were our major takeaways. So now I want to dive into some of the data that supports our opinions on these recommendations. And the first is really driving home how correlated your organic matter measurement and your POC measurement are. This makes a lot of sense. Both of these, to some degree, are looking at that carbon content of the soil. And you can see that across all of our fields, they track together. Our R value was about 6.6. So that's a really, when we're talking about things when we're in a noisy field situation, that's a pretty good R squared value for that correlation. So generally, they're going to be telling you the same things about the trends in carbon in your soil. Really, at the end of the day, I think for soil health, one of those primary goals is going to be to impact our yields in a positive way on the farm. And so here I'm first showing you our trend lines between organic matter and both corn and soybean yield. Corn is on the left and soybean is on the right. You can see soybeans appear to be a little more responsive to increases on organic matter than corn, but both are positive. This makes a lot of sense. This is why us folks in Ohio with those lower organic matter soils are jealous of our neighbors to the west that have those beautiful black prairie soils. Intuitively, this was a trend we expected to see, and it's always great to see what you expect in your data. POC is the same story. So these are the same charts, and you can see that as our POC values increase, so to both our corn and soybean yields, interestingly, to a slightly lesser degree here, but still positive. So if yield is your ultimate goal, I do think both of these tests can be informative. The next part, thank you, Jim, for clarifying that in the comments, that POC does stand for, I can't even pronounce the permanganate oxidizable carbon. So switching to using these tests to detect changes in soil health due to management. So the next series of slides, I'm going to be showing you the data that the results from the data looking at our management practices across all of the fields that we sampled. So the way that we tried to understand the question of how the time component of these practices impact soil health is, again, by that broad sampling and then asking farmers whose fields we sampled how many years each one of those fields had been under a particular management regime. So in this case, we're looking at continuous no-till. You can see we have quite a few fields that are not no-till. So they have 0 years of no-till as a practice. And over time, you can see our greatest number of years in no-till was 50 in this data set. And we've got that whole range. So this gives us, in a way, the ability to see what kinds of changes we would expect in these soil health values over time, but just in a time point snapshot today. So overall, the story for continuous no-till isn't the rosiest in terms of soil health. You can see POC is on the right and organic matter is on the left. Both of those have a slow but overall negative trend over time. I don't think this was particularly surprising when we looked at this data. We know that, again, we're looking at that 8-inch core, and the longer you're in no-till, the more you're going to be concentrating that organic matter buildup in those top few inches of your soil. So it's not overall surprising. We did still see a negative trend in those shallower depths, however. You got to believe me because I'm not showing you the data. But I do think with our other management practices, we see some promising results as well. So again, this is the same style of data where we're showing you the number of years each of these fields was in cover crops. You can see we had a lot more fields that had long-term no-till than we had cover crops. You can really see cover crops caught on somewhere about 10 years ago because a lot of our data falls in that 0 to 10 year range. But we do have some fields that were either in a perennial system or had a longer term rotation with cover crops. I think here the positive story is we do see with POC and organic matter both an increase over time with cover crops being inserted into the system. So the story of cover crops bringing in organic matter, bringing in carbon to your system and stealing it away in your soils is absolutely accurate according to our data. These two graphs really drives home when I say that POC is more responsive over time. Overall, we know at the end of the day, these fields are the same. So the only difference in these two graphs is the way that we are measuring that real change or how we're trying to estimate that real change. You can see that the positive trend with POC seems steeper. So that's what indicates to us that this test is going to show that impact more quickly on paper through that test. So organic matter, if you're willing to wait, is going to tell you the same story as POC. But if you want to see changes quickly, POC is see those changes translated into data more quickly. POC is going to be that better option for you. To close out the data here, I'm going to show you that story from the other three with our manure applications. So again, same thing, years of manure application to a field. We can, to a lesser degree than the cover crop, but still a positive trend with building organic matter in our soils over time, building carbon in our soils over time, as indicated by these two tests. So I think promising if your goals, we've now talked about, if your goals are yield, we saw a positive correlation with these tests and increased yields. So if you want to increase yields through soil health, I think there's some options here for you. If you want to be able to track changes, again, I think both of these tests are excellent options. So what does that mean when we take this home to your farm? again, circling all the way back, when we are wanting to incorporate soil health testing into your farm, my number one recommendation is to think about what your goals are and how you want to use tracking soil health in your operation. Once you determine that, I think it's going to fall into place which tests make the most sense. And the things that might matter to you are going to be potentially cost, the convenience. Can you just send these tests in and get reliable results on the same cycle that you're sending in your regular fertility tests? And then the reliability of those results. How careful do you need to be with those samples? Those are going to be the drivers that help you select what tests are going to match best with how you're going to handle them, how you're going to collect them, and what's going to answer the questions you're looking for. And really then at the end of the day, we really saw in that data that it takes quite a few years to start seeing things move in that direction that you're hoping to move by increasing soil health. So the real key here is being patient. That change is going to take time. And as that change is happening, it's also going to take time to detect it in the results you're getting on your health tests. My final slide is if you're interested in what we're doing with eFields, the summaries of the soil health tests over the last 5 years are in those reports, but there's also a whole slew more data that you can check out that's on farm based and applied in nature. So you can scan that QR code on your phone and that'll take you to the reports. But I'm excited. I've seen some things popping up in the chat, so I think we've got some good questions from you guys.
Thank you very much, Elizabeth and Jim, for this great presentation. I learned a lot, as I always seeing from modeler side, how this field varies and need to consider in our model aspect. We are getting the really good conversation through chat box here, and we have a couple of questions. So first, I think we are fresh here from Elizabeth's presentation. We have a question on some graph. So on the toxicity and organic matter graphs, how did texture affect the slight negative trend? Typically, the longer no-till sites tend to be an early adopter with soils that were prone to higher rates of erosion.
Vinayak, I'm going to ask that you give me a timeout on the texture question. I've only had half a cup of coffee, so I'm going to pull that data up so that I don't mislead you. I do think you're correct in your assessment that our no-till farmers are our more conservation conscious folks. And so I think this is really an important story for them to hear and reflect on. No-till is great at achieving the goals that a lot of them have previously set out. Originally, we were looking to reduce erosion, keep our soils from moving where they weren't supposed to, lock in that residue, and not break it down as quickly. And I think no-till has achieved those goals. But when we start talking about soil health, I think our goals are a little bit different. And I think this is a great opportunity. Our data points to some great strategies to reverse that negative trend. The no-till cover crop combo is still going to lock in those original goals of no-till, of stopping erosion or reducing erosion, but that it's going to add in that next benefit of building back. I think we know that a lot of those cover crop fields are also no-till. So I think it's interesting that that shift in the data is so strong that we see cover crops very quickly turn that trend around. So I think that's a great success story in our data for cover crops and demonstrates that progressing in our conservation practices is really important too. So if you're a no-till farmer and you're sad about the data that I just shared, I think cover crops might be your next logical step to continuing down that. I mean, really, it's a path, right? Continuing down the path of conservation on your farm. And I'm going to pull up that data, Vinayak.
Thanks, Elizabeth. Next question we have, can you compare and contrast tests for soil dispersion and what is used to obtain potential mineralization nitrogen in the soil health testing framework? I think this is for Jim.
Yeah, okay, so part of the question was comparing the different respiration tests that are out there. Okay, so yeah, there's a good handful of respiration tests that are out there. And so, well, this was maybe about 4 or 5 years ago. I was part of the Soil Health Initiative or Soil Health Institute's North American project, and part of that project did quantify different respiration tests to see which one would be, quote, the best in terms of quantifying respiration. And that study, and it's been published, I think it was published in 2021, it showed that 24-hour CO2 burst was basically telling you maximum respiration as compared to, like, you could run a 4-day test, you can run a 2-day test. Sometimes people don't even use respiration. So respiration isn't used in soil management assessment framework. We just use microbial biomass carbon as an indicator, but respiration, 24-hour respiration is good. If you use 48 hours, you'll probably get you'll get the same result. What was the second half of the question?
So what is used to obtain potential mineralizable nitrogen in soil health testing framework?
Okay, so that one's all over the board. And so in the soil management assessment framework that I use in my program, we use something called potentially mineralizable nitrogen, which is, it is painful. It's a 28-day aerobic incubation. And so what you do is you measure nitrate and ammonium in the soil at the beginning, and then you incubate the soil. The soil's typically moistened, and you incubate it over time. In my case, it's the program we use, it's 28 days. Some people use 7 days and it's anaerobic, but it's still an incubation. And then you measure nitrate and ammonium in the soil after the incubation. And so the difference is how much nitrate and ammonium was released or mineralized from the organic fraction. So that's one way you can do it. Another way that you can do it, and Elizabeth, you talked a little bit about this, you can measure ACE protein, all right? So ACE protein it stands for autoclaved citrate extractable protein. And it's complex, but it's basically a way to extract organic nitrogen from a form that would be readily accessible by microorganisms. And the test isn't very difficult, and you don't have to incubate anything, which is attractive. And then there's a third way that you can do this, and I'm drawing a blank, but there's either way, it's not like, you can stick a test strip in a soil and get a number. It's not that simple.
We have one more question in addition to that. Additionally, how does POXC compare to, I don't know if it's pronouncing right, POM, what is the long form of that, for testing the active fraction of soil carbon?
Elizabeth, do you have a good feel for that?
I do not. I was trying to look at that question, to try to find that question, to read it and see if I could. While we pause and think about that one, Vinayak, your answer to how texture impacts, the general trend is our finer textured soils trend towards higher both organic matter and POXC, which makes sense. If you think about our coarser soils are typically those sands. We associate sands with most typically a lower organic matter content, a lower carbon content. So that trend intuitively makes sense. But that is important to keep in mind overall when looking at these results. If you're on a sand, you're probably not going to see a rapid or dramatic increase in organic matter, and your baseline in general is going to be lower when you're starting out.
Thanks, Elizabeth.
Yeah, I saw you
James.
put the question in the chat there, Asmita.
Yes.
So how does POXC compare to POM? So POM stands for particulate organic matter. And I don't know the answer off the top of my head, but I can give you an educated guess. And so particulate organic matter is the organic matter that's associated with soil particles, mostly oxides like iron oxides. It's typically organic phases in the soil that are protected against degradation from microorganisms, right? Because it's associated with mineral phases that sort of take that organic phase out of and away from microorganisms that can attack it and degrade it. And so POXC theoretically is completely different. All right. So the initial idea with POXC, which is permanganate oxidizable organic matter or organic carbon, I'm sorry, is the fraction of carbon that can be theoretically degraded by microorganisms, so it's active, and it likely is more associated with PAOM, so particulate. I'm sorry, with MOAM, mineral. I went backwards. Oh my gosh, I am like Elizabeth. I need more coffee. Okay, disregard everything I just said, and let's start over.
Thanks. That's
I
good.
really
Yeah.
need more coffee.
Well, okay, so I think we have a question on like fertility recommendation. Can soil health tests help update fertility recommendations? And will the future tri-state recommendation will be updated to include some credit for good soil health or nutrient credit from cover crop biomass?
This is a tricky question. We're still sorting through the data. One of the aspects I've most recently started to look at is that fertility interaction with soil health. Right now, I've looked at management practices in fertility, and in our data, we're not seeing an increase in extractable nutrients in our soil over time in any of those scenarios. So I think obviously there's a lot more to learn here, but I'm not sure that we are going to see the payoff that we're hoping for in terms of some of these conservation practices, especially if, like, if you really think about it, if you are increasing yields, the increase in yields is likely driven to that increase in available nutrients. And if the increase in available nutrients is being used by our crops for yield, we're not building those values over time. And I think that's what we're seeing in our data is that benefit is immediate to your crop and you're still needing to feed over time to maintain that level of productivity. Again, I still have a lot more to dig into with that data, so maybe that's a 2026 webinar. But at this point in time, I don't think we have enough data to be really responsive or to change our overall recommendations based on what we've learned from soil health testing so far.
Thanks, Elizabeth. I think we covered all the questions. I have a couple of questions for you guys. So,
Before
not
you ask, can we go back to the
yes, sure, yes.
POM? Yeah. So I put something in the chat about the correlation. There's likely a correlation between POM and epoxy, just because they both can be degraded theoretically by microorganisms. But there's a caveat. There was a recent paper that came out from scientists at Oregon State University that looked at POC and what it was really telling us. And it was an interesting paper because it showed that POC, initially we thought POC was the fraction of organic carbon that could be utilized by microorganisms. But this paper showed that POC is actually related to probably the types of plants that are present in an agroecosystem. And so, yeah, it's related to microorganisms, but it's even more so related to like plant and plant detritus or plant root exudates that come from what you're actually growing or raising. And so you can think about this idea that maybe it's related to biodiversity in agroecosystems. Just FYI.
Thanks, Jim. I think one like a last question from my side. Like, let me see if there is any question. Okay, we have more question from audience, so I think we can cover that. Do you have any data on the influence of industrialized biofertilizer on soil health over time? If you do, does this data show a positive influence or a negative effect?
With biofertilizers.
Yeah.
Oh, what do they mean by biofertilizers? Just out of curiosity.
Moma, do you have, Rachel, can you maybe allow her to mention about those, like industrialized biofertilizer?
Oh boy. Yeah, I still don't know. Go ahead. Is that Mama?
Yeah.
while she's coming online, I know that, one of our last sessions, we had somebody, his name was Brian Arnall from Oklahoma State University, and he wasn't talking about bio fertilizers, but he was talking about biostimulants and not seeing a response. And so more often than not, he doesn't see a response with biostimulants in his program, which means he wouldn't see a response in soil health either. But I don't think that's the question. If I was going to say anything about anything organic, like manures, compost, etc., yeah, you would see a positive, typically you would see a positive response. Oh, with like maybe biochar. Yeah, and biochar depends. It depends on what the feedstock is. So if you make biochar out of wood-based materials, you're likely not going to see a great, if any, response in terms of soil health because it's mostly carbon and it's recalcitrant. But if you make biochars out of a manure or another organic, an amendment that has high nutritive value, you're going to see, you likely will see a response in terms of soil health.
Thanks, Tim. I think in meantime, Ryan is putting the slides again for concluding slides. I just have one question about like there are some mobile app is coming for the soil test, like a quick results through mobile phones, like an app. Do you have insights on that?
That would be really cool. The only app that I know of that I sometimes use is the Slakes test, and it's for determining aggregate stability. Basically, you can use it in the field, but you can also use it in a laboratory. You can use it on your kitchen countertop, to be honest with you. You use your phone and it sort of takes pictures or video over time and it looks at the degradation of aggregates when they're being wetted. And sometimes the app works well for me and sometimes it goes belly up. And so I haven't used it in a while. I think it's been updated. I don't know if there's an actual soil health app that's available because you still need to quantify somehow physical, chemical, and biological. I can tell you that one of the programs that I'm currently working on and others are is actually quantifying soil health using an online tool that's not quite out there yet, but it's a form that you fill out to describe your management practices and what you physically see in the field. And so the idea is to get away from soil health testing altogether and use a form. And so I have a big project looking at this across 6 states, quantifying soil health like I share with you and Elizabeth share with you in the slide deck, but also using a field form that producers would fill out and then eventually, and give me 5 years, and we're going to correlate all the data to see if the field form actually tells us what the laboratory tells us. And can we get away from lab testing?
Same. Just Vinayak mentioned there is a app, Soil One is based on the Poxy, again, based on the field test kit. I think it's soil1.com. Yep. Thanks.
The issue with the field test kit, in my opinion, is it works pretty well. But the data, like I've seen some of the data from across the state of Ohio, and you have to take the data, like any data, with a grain of salt, not just Rafiq's data set, but even the SMAF, the CASH, the Haney test, because the values are only as good as wherever you're located. So you might get a low soil health test score, but there's no way to improve it. And the low soil health test score is actually the highest score you can get in an ecosystem. We can talk about it later if you want.
Thanks, Jim. Thank you very much, Elizabeth and Jim, for today's presentation, and it was a good conversation. Ryan, we'll go through on some concluding slides.
Thanks, Asmita. Can't turn my camera on, but I'll just go through these slides here. For anybody needing the CCA credits, you can either scan that QR code or you can go to this website here that is in the blue for one of those credits. So that is there. For next month, you can join us again on April 12th for the Soil Health and Urban Sphere. And for next year, if you have any suggestions on topics, you can email Smita or Mambir at those two email addresses listed down there at the bottom. Elizabeth mentioned it, but the 2024 eFields report's available. You can either go to go.osu.edu/eFields, or you can scan that QR code. There's a lot of great information in there across the board for those agronomic crops. And then just want to say thanks everybody for attending. We had a lot of great discussion, good presentations, and great questions. If you have any questions about today's webinar, you can email either Asmita or me at those email addresses there listed on the screen. And again, thanks everybody for coming.